Hey everyone, how's it going? In today's video, we're going to be learning about the signaling pathway for these two messenger molecules, epinephrine and norepinephrine. Now, in our last video, we learned about the signaling pathway of a messenger molecule called dopamine. Now, dopamine is a type of messenger molecule called catecholamines. So, catecholamines is this general, umbrella of signaling molecules, and one type of catecholamine is dopamine. However, epinephrine and norepinephrine, these are also types of catecholamines. So norepinephrine and epinephrine, these are also catecholamines. So in the last video, we learned about dopamine. That's catecholamines part 1. This video is catecholamines part 2, and we're going to learn about norepinephrine and epinephrine. So let's get into it. So before we get into epinephrine and norepinephrine, we first should review what catecholamines are. So as I said, catecholamines is an umbrella term for a bunch of different messenger molecules. And catecholamines, basically, if you look at the name, you can break it down into two parts, catechol and amine. So catechol, the first part, basically means that they are structured around this catechol structure that you see here. So this catechol structure is the backbone for all catecholamines. And then the second part, amine, basically means that it has an amine group. An amine group is basically when you have your chemical, and then your amine group is where you have a nitrogen group bound to a couple of hydrogens. So that's your amine group. So that's catecholamines. Catecholamines is when you have your catechol structure backbone, and then you have an amine group. And lots of different messenger molecules fall under this category, such as dopamine, norepinephrine, and epinephrine. And we're going to focus on these two messenger molecules in this video. And specifically, when I say that we're going to learn about them, what we're going to do is we're going to look at the signaling pathway for these messenger molecules. So we're going to look at how epinephrine and norepinephrine get produced, then how they get packaged, how they get released, and then how they go and produce responses in your body, and then eventually how these responses can get terminated. So let's get into it. Now, before we get into the signaling pathway for norepinephrine and epinephrine, I want to say that I've already made a couple of videos fully dedicated to packaging mechanisms, common release mechanisms, and common termination mechanisms. I'll link all three of these down in the description below, but I'd really recommend watching these videos first to give a bit of foundation before getting into this video. And I'll also really recommend to watch my first video about dopamine, the part 1 of catecholamines, before getting into this one. But with that said, let's see how epinephrine and norepinephrine get produced. Now, in our last video, we looked at the catecholamine production pathway, so how catecholamines get produced. And again, I'd really recommend watching the previous video, Catecholamines Part 1, as it will explain this process much more in depth, and this process is very relevant for the messenger molecules we're going to look at today, which is epinephrine and norepinephrine. But going over it briefly, in our blood, we have a messenger molecule called phenylalanine. A hydroxylase enzyme comes along and adds a hydroxyl groups to your phenylalanine, which turns it into tyrosine. Tyrosine then gets transported from your blood into a neuron. And then in your neuron, it again gets hydroxylated. So a hydroxyl group comes and attaches to it. And this turns it into L-DOPA. So L-DOPA is an amino acid, meaning it has a amine group and a carboxylic acid group. Then an enzyme called decarboxylase comes and it removes a carboxyl group. So then you only have your amine group, and as a result, you go from an amino acid to an amine, specifically a catecholamine, because you have your catechol structure, and now you only have an amine group without your carboxyl group. And that gives you your messenger molecule, dopamine. So now that dopamine has been produced, it has to get packaged, so we can transport it inside this storage compartment known as a vesicle with the help of your enzymes called VAT and VMAT. What VAT does is it basically pumps protons, positively charged protons, into your vesicle, generating a force called the proton motive force. And then your second enzyme, VMAT, it can exchange the positive protons inside your vesicle. It can exchange them for positively charged, let me make that a different color, for positively charged dopamine. So it pumps out protons and it pumps in dopamine. And this process results in dopamine being stored inside your vesicles. So this is how we produce and package dopamine. But now that we've produced and packaged dopamine, we can now produce epinephrine. Let's take a look at how this works. So now that we have our dopamine inside our vesicle, we can now continue this catecholamine production pathway to produce norepinephrine. So another hydroxylase enzyme comes along and adds a hydroxyl group. It adds an OH, and that turns dopamine into norepinephrine. And here it might get a bit confusing, but norepinephrine actually has an alternate name, which you may have heard of, called noradrenaline. Oh, noradrenaline. So norepinephrine and noradrenaline are the exact same thing, just two different names like British English, American English. But adding your hydroxyl group to your dopamine converts it into norepinephrine / noradrenaline. And now we've produced norepinephrine or noradrenaline, but the cool thing is we haven't just produced it, we've also packaged it. Remember the first two steps of our production pathway, of our signaling pathway? We have production and we have packaging. We've just covered both of these steps simultaneously because, look, we've already packaged our dopamine inside our vesicle by transporting it into our vesicle with the help of our VAT and VMAT enzymes. So dopamine's already packaged. So then when we turn dopamine into norepinephrine by adding this hydroxyl group, now not only have we produced norepinephrine, but this norepinephrine is also packaged inside our vesicle. So we've killed two birds with one stone. Our norepinephrine is packaged, it can be stored and stored until later when it's really needed, and then when it's needed, it can get released from the neuron and go out to produce responses. But one thing to consider is the question of when does this catecholamine pathway stop here at dopamine, and when does it continue on to produce norepinephrine? Because some neurons, they stop here. After you produce dopamine, then you don't add a hydroxyl group. You don't go on to norepinephrine. You just stop here, dopamine is stored, and then eventually dopamine gets released and produces responses. But in other neurons, you don't stop at dopamine. You add the hydroxyl group and you go on to norepinephrine, and then this is what gets stored and eventually gets released out of the neuron to go and do responses. Well, it actually depends on the type of neuron. So dopaminergic neurons. These neurons, as the name suggests, they will store dopamine and release dopamine to go do responses. Whereas noradrenergic neurons are different types of neurons, and these, as the name suggests, they don't stop at dopamine, but they go on to noradrenaline or norepinephrine. So really it depends on the type of neuron, and so the different types of neurons can store different types of messenger molecules, and then when they're released, they can go off and do different responses. But that's how norepinephrine or noradrenaline gets produced and packaged. So that covers the first two steps for noradrenaline. Now let's look at how noradrenaline gets released out of the neuron. Okay, so now that your norepinephrine is packaged inside your vesicle, it has to undergo the process of release. So it may not be released immediately. It can be stored inside this vesicle for some time, but when the time is right and we want norepinephrine to be released so it can go and produce a response, then it has to be released out of this neuron somehow. And this involves these 4 steps. As you can see, I've drawn these little proteins hanging off your vesicle and hanging off the membrane of your neuron. So these proteins hanging off your vesicle are called VAMP or V-snare proteins. And these proteins, which are, these proteins, which are hanging off your the membrane of your neuron, they are called T-snare or SNAP proteins. So you have VAMP and SNAP proteins, and these proteins are critical for the release process. So the first step, translocation, is when your vesicle moves over closer to your cell membrane. So it makes its way over, and then after translocation comes tethering, which is when your VAMP proteins and your SNAP proteins, they link up together, they attach. And then after they attach, more interactions happen between your VAMP and SNAP proteins. And that's the step of docking and priming. It primes itself for the final step of release. And then in this final step of fusion and exocytosis, what happens is your vesicle membrane fuses together with your neuron membrane. And when this happens, it looks something a little like this. So your vesicle comes over, and all the VAMP and SNAP proteins interact, and it goes onto your cell membrane, and then slowly it starts to fuse together, and it looks something like this. Like that, it starts to fuse together. And then, eventually, it's going to open up even more, and then it's going to start to look like this. It's going to open up, and then the norepinephrine that was inside your vesicle is going to be able to leak out. And that is the final step of fusion and exocytosis. So fusion is the fusion of your two membranes, your cell membrane and your vesicle membrane, and then exocytosis is the release of norepinephrine out into your synapse outside of your neuron. So that's the release mechanism. So I know that release is a bit complicated. This is a pretty complicated process that we kind of rushed through. Don't worry, as I've said before, I have a full video that is fully dedicated to release mechanisms. I've linked it down in the description below, and I'd really recommend watching that. It'll give a good background for understanding this release mechanism a little bit better. But with that, we've successfully covered the third release step of norepinephrine. So now norepinephrine has been released from our neuron and it's out into the synapse. Okay, so now that norepinephrine and noradrenaline has been released from your neurons and it's out into the synapse, now it can go on to different receptors to produce responses. Now, norepinephrine or noradrenaline, remember they're the same thing, they can bind onto a couple of different types of receptors. There's alpha receptors and beta receptors, and they can be further classified. There's alpha 1 and 2 receptors, and then beta receptors can be beta 1, beta 2, or beta 3 receptors, but for the purposes of this video, we are going to focus on beta receptors altogether, but separate alpha receptors into 1 and 2. Now, these receptors can be found on a variety of different cells, and norepinephrine can go and bind onto these receptors to produce different responses. And the way that these receptors work is a little bit different. So this diagram should help explain things. So your norepinephrine or noradrenaline can go and bind onto alpha 1 receptors or alpha 2 receptors or on beta receptors. Now, for the purpose of this diagram, I've shown all these receptors right next to each other. In reality, these receptors can actually be found on a variety of different cells, not necessarily all in the same cells, but this is just for the purposes of the video. So as you can see, these different receptors, they're all GPCRs, meaning that they are linked to these intracellular G proteins. Now, if you haven't watched my videos on G proteins, specifically GI, GS, and GQ proteins, I'd really recommend watching all those videos. I'll link all of them down in the description below. They'll definitely help understand this a lot more. But as you can see, your alpha 1 receptors, these are GPCRs that are linked to GQ proteins. So when norepinephrine goes and binds to your alpha 1 receptors, it activates GQ proteins. And GQ proteins, when they're activated, they go and activate an enzyme called phospholipase C. They increase phospholipase C activity, and phospholipase C produces messenger molecules IP3 and DAG. And these second messengers, IP3 and DAG, can go off and do various responses inside the cell. So that's your alpha-1 receptor, but norepinephrine can also go off and bind to your alpha-2 receptors. And your alpha-2 receptors, instead of being associated with GQ proteins, they're instead associated with GI proteins. GI proteins, they lower the activity of your enzyme called adenyl cyclase. On the other hand, norepinephrine can instead go and bind to beta receptors, and beta receptors are linked to GS proteins, which also act on adenyl cyclase, but they increase adenyl cyclase activity. So these two different types of receptors, alpha-2 and beta receptors, they both activate different G proteins which both act on adenyl cyclase, but one lowers response and the other one increases response. And adenyl cyclase, it produces a second messenger known as cAMP, which can produce responses inside the cell. Just like how IP3 and DAG can produce responses inside the cell, these are just different types of second messengers that can produce different types of responses. But basically, your alpha-2 and beta receptors, they can kind of counteract against each other. Since one lowers adenyl cyclase activity, the other one increases the activity, they counteract each other to strike a balance in overall adenyl cyclase activity to keep response under control. But this basically summarizes the three different types of receptors that norepinephrine or noradrenaline can bind onto. Again, they're all GPCRs, but they're linked onto different G proteins. And when they activate these G proteins, they can activate or deactivate different enzymes and produce different second messengers that result in different responses. Now, one important thing to note is that the responses of norepinephrine are very closely linked with your sympathetic nervous system. So your nervous system has this section, these two different systems called the parasympathetic and the sympathetic pathways. So the parasympathetic nervous system is basically your rest and digest states, whereas your sympathetic nervous system is, you probably heard of this before, flight or fight. So when you're in danger, like, let's say you see a lion or a spider or something, or, you're getting attacked, you have to run away or fight back. That's your sympathetic nervous system. Flight or fight. You run away or you fight back. When you're just chilling and, your body's in rest mode and you can digest food, that's your parasympathetic system. Rest and digest. So norepinephrine, its responses are very commonly linked with this sympathetic part, with the sympathetic nervous system, the fight or flight pathway. So a lot of these responses, when norepinephrine goes and binds onto alpha and beta receptors and produces different responses, a lot of times these responses are parts of your sympathetic pathway. Now, don't worry too much about this for now. In a future video, we're going to go more in depth about the parasympathetic and sympathetic nervous system, and when we do, we'll go a bit more in depth into these responses. But for now, just know that norepinephrine can bind onto these different types of receptors, activate different types of enzymes and produce different responses, and know that norepinephrine is very heavily involved in the sympathetic nervous system, which controls your fight or flight responses. So this whole time we've been talking all about noradrenaline and norepinephrine. But if you remember, this video is not only about norepinephrine, it's also about epinephrine. So what is epinephrine? How is it different than norepinephrine? Well, you see, when your norepinephrine is released from your neuron and it's out in your synapse, then your norepinephrine and adrenaline, it can either go and bind to your alpha or beta receptors and produce responses as we discussed, or it can go over to something called the adrenal medulla. The adrenal medulla is a part of your adrenal glands. Adrenal glands are these two glands that sit on top of your kidneys. And the adrenal glands is made of the adrenal cortex and the adrenal medulla. So the adrenal medulla is a part of this adrenal gland. So your norepinephrine and noradrenaline can go over to your adrenal medulla, and it can basically get methylated. So an enzyme called methyltransferase adds a methyl group to your noradrenaline or norepinephrine, and that turns it into epinephrine or adrenaline. So it turns norepinephrine into epinephrine, or it turns noradrenaline into adrenaline. Just like how norepinephrine and noradrenaline are two names for the same thing, epinephrine and adrenaline are also two names for the same thing. And they are created by adding this methyl group to norepinephrine and noradrenaline, and this happens in the adrenal medulla. And so what's the difference between your norepinephrine noradrenaline and epinephrine adrenaline? Well, they're both messenger molecules that can go off to do similar responses. They're both involved in your sympathetic nervous system. But the main difference is that norepinephrine and noradrenaline are neurotransmitters, whereas epinephrine and adrenaline are neurohormones. But what does this mean? So in my first ever video where I talked about the general cell signaling pathway, I broke down the differences between different types of messenger molecules, such as hormones versus neurotransmitters. I really recommend watching that video first. I've also linked that down in the description along with all the other videos, and I really think that that could help with this. But just to go over it briefly, I think that this table can really help break down the differences between a neurotransmitter such as norepinephrine and a neurohormone such as epinephrine. Also keep in mind that norepinephrine is the same thing as noradrenaline and epinephrine is the same as adrenaline. But basically, a neurotransmitter is a messenger molecule that is stored in a neuron and then it gets released and it travels across a synapse and binds to receptors on your postsynaptic neuron, which comes after your synapse, and produces responses. The synapse is a tiny, tiny gap between these two neurons, or this can also be between a neuron and another cell, such as a muscle cell or a gland. But the point is, you have this tiny, tiny gap in between these cells. On the other hand, a neurohormone is a messenger molecule that is, comes from a neuron. But instead of going over a tiny, tiny gap to get to your next cell, it has to travel a much further distance. And in fact, it travels through the bloodstream. And it travels a far distance through the blood to get all the way to this tissue, which is far, far away. So neurotransmitters, they travel across this tiny synapse and for very short distances, whereas neurohormones have to travel through the bloodstream and for much longer distances. So because neurotransmitters are traveling over a short distance, they don't have to be as resistant and strong because, they're just kind of going over to the cell next door. So as a result, they're less stable. On the other hand, neurohormones are traveling a much further distance, so they have to be able to resist a lot more wear and tear without breaking down. So neurohormones are a lot more stable. So that's the main difference between norepinephrine and epinephrine. They are designed for different things. If you got to send a messenger molecule over just a short distance from a neuron across a synapse to your postsynaptic cell, then you keep it as norepinephrine because it's a neurotransmitter and it's perfectly designed for this. But if instead you need to produce a similar type of response but in a cell that's far away from your neuron and it needs to travel through the blood, then you can't just stay as norepinephrine. You got to convert norepinephrine into epinephrine via methylation at the adrenal medulla. So if you need more long distance communication, you can convert norepinephrine into epinephrine via this process. And now all of a sudden, you have epinephrine, which is a neurohormone and is able to travel through the blood much further distances, and it's a lot more stable. So this way you're able to get your signal to your destination, which is much further away, without it getting broken down. So it depends on your situation. In some cases, you want a short-term messenger molecule, and in other cases, you want a long-term messenger molecule. But generally, these both norepinephrine and epinephrine do very similar responses. They're just applicable for different scenarios. So with that, we've covered the response step of epinephrine and norepinephrine. Both pretty similar responses, but used in different scenarios. But at the end of response, the response must be terminated to prevent it from getting out of control. And there's a few different ways that these messenger molecules can be terminated. Now, I know I've said this again and again, I'm sorry, but I've already made a video dedicated fully to termination mechanisms, and it's really going to help understand this video. We can't go that much in depth or else this video would be an hour long, so I'd really recommend watching that video first or referring to it if you want a better explanation on any of these concepts. But these are your three main termination mechanisms for your norepinephrine. So norepinephrine can be reuptaken into cells and stored there for later. It can be degraded or converted basically inactivated by various enzymes to stop it from doing a response, or the receptors that it binds to can be desensitized. Let's briefly look at how each of these mechanisms work. So the termination mechanism of uptake, as we said, it's when your norepinephrine is released into your synapse, and then if you have excessive response, you can terminate and control this response by having your norepinephrine get uptaken into cells and stored for later. So there's two different types of uptake. There's neuronal and non-neuronal. As the name suggests, neuronal uptake is when your norepinephrine gets uptaken into neurons, either the neuron that just released it or some other neuron that's nearby. And non-neuronal uptake is when your norepinephrine gets uptaken into some other non-neuron cell, so let's say some tissue cell. And in doing so, they're basically pumping the norepinephrine back into a cell, and then it can be stored until it's really needed. And by that, they can prevent the amount of norepinephrine outside of your neuron, inside your synapse, from being too excessive, so they can keep responses under control. Now, for the process of neuronal uptake, so in order to pump norepinephrine back into a neuron, you have the help of this transporter called NET. So NET is your transporter present on your neuron cell membrane, and it assists with the process of pumping norepinephrine back into your neurons in order to be stored. Another important thing to note is that these processes of uptake into either your neuronal or non-neuronal cells, these reuptake mechanisms can be inhibited by various different things. And by inhibiting these reuptake mechanisms, then they result in more norepinephrine being in your synapse and more responses. So they increase response basically by preventing termination. So things that can increase response for neuronal, let me write that in blue. So things that can inhibit neuronal reuptake are, for example, cocaine, antidepressants. What else? Amphetamines. On the other hand, things that can inhibit reuptake into non-neuronal cells include steroids. So taking these things prevents norepinephrine from being reuptaken into these cells, and therefore they increase the amount of norepinephrine you got in your synapse, and they increase the amount of norepinephrine that can go off and bind to receptors and produce responses. So that basically covers uptake. The next way that norepinephrine can get terminated is through enzyme degradation. So different enzymes, specifically MAO and COMT, can convert or degrade norepinephrine in order to basically deactivate it. And when norepinephrine is deactivated, it can no longer go onto receptors and produce responses. So the first termination enzyme is MAO, which stands for monoamine oxidase. And what monoamine oxidase does is, as the name suggests, it oxidizes your norepinephrine, and in doing so, it basically inactivates it. Now, MAO is not limited just to norepinephrine. It can act on norepinephrine. It can also terminate dopamine as well as serotonin. Now, MAO is made in the mitochondria of different organs, such as your liver and the epithelium of your intestine. And what's interesting is, just like with reuptake, different things can inhibit your MAO enzyme, such as antidepressants. So they can prevent MAO from being able to oxidize and terminate your norepinephrine or your dopamine or your serotonin. And in doing so, it actually increases it increases the presence and it increases the responses of your messenger molecules. So MAO, monoamine oxidase, it oxidizes these messenger molecules in order to terminate them, and it gets inhibited by things such as antidepressants. So that's one termination enzyme. The second one is called COMT. So the second termination enzyme, COMT, stands for catechol-O-methyltransferase. As the name suggests, catechol means that it acts upon catecholamines like norepinephrine, epinephrine, dopamine, and methyltransferase means that it acts by adding a methyl group. So it adds a methyl group to your norepinephrine or your dopamine or whatever other catecholamine you're dealing with, and in doing so, it terminates it and it prevents response from happening. And COMT is found in a pretty big variety of cells in your body. And that basically covers your second step of degradation. You have two enzymes, MAO and COMT, and both of them can act upon your norepinephrine or other messenger molecules such as dopamine, serotonin, and in doing so, they terminate them. Now, the third termination mechanism is called receptor desensitization. So, of the three termination mechanisms, the third one of receptor desensitization is undoubtedly the most complicated, and there's just no way that I can fully go into it in this video. So as I said before, I made a video fully dedicated to termination mechanisms. I've linked it down in the description below. If you want to properly learn about the steps and the pathway of receptor desensitization, I really recommend watching it specifically at timestamp 12 minutes and 4 seconds. That's where I start talking about receptor desensitization and I go into it really in depth. I'm sorry, I really like to, go through all of these steps in depth in one video so that it's nice and clean. But if I did, the video would just be more than an hour long, and it really helps to understand the base fundamental concepts before applying them to specific messenger molecules such as dopamine or norepinephrine. So again, I really recommend watching that video first, but just to go over it briefly, receptor desensitization is a bit different from your first two termination mechanisms, whereas uptake of messenger molecules and degradation of messenger molecules, they both act on your messenger molecule. But receptor desensitization, it actually doesn't do anything to your messenger molecule. It instead acts on the receptors to which your messenger molecule goes and binds to. It inactivates these receptors to keep responses under control. So here in this diagram, you have norepinephrine being released from one neuron, and then it goes over to this neuron, and it can bind onto all these receptors. And by binding onto all these receptors, it can produce responses inside your cell. But what if you have a lot of norepinephrine, and it's going to go and bind onto all your receptors and produce a huge amount of response? It's going to produce too much response. Remember, we don't want too much response. We want to be able to control the response. But what receptor desensitization does is basically it inactivates some of these receptors by pulling them inside the cell. So it internalizes some receptors. So now these receptors are inside the cell. Sorry, it's a little crammed in here, but they're inside the cell instead of outside the cell. So now instead of having, let's say, 10 receptors outside, you only have these 5 left. You've pulled the other 5 inside. So because there's less available receptors for norepinephrine to go and bind onto, the total response inside the cell is decreased, because now you have the response of only 5 receptors instead of 10 receptors, because the other 5 are inside your cell now, and norepinephrine cannot go inside to bind onto them. And this is called receptor desensitization, and it helps keep responses under control. Now, after some time, these receptors can be returned back up to the surface. They can kind of be recycled, and that's called resensitization. So they can be returned back up to the surface. So now the cell is back at its original 10 receptors. I know this is not actually 10 receptors, I'm just saying 10 and 5 to keep it simple, but now you're back to your full receptor count. But let's say you still have a lot of norepinephrine, and it's coming and binding onto all of these receptors and producing too much response again. Well, then the cycle can repeat. So some other receptors can be desensitized and pulled into the cell. So now you're losing all of these receptors, and those receptors are inside the cell, and the cycle can continue. So it's a cycle of desensitization and resensitization, pulling receptors into the cell to deactivate them, and then recycling receptors by returning them to the cell membrane, and together desensitization and resensitization create a delicate balance to keep overall cell response under control. And that's the general idea of receptor desensitization, very simplified, and if you want to learn more, be sure to check out my termination video at timestamp 12 minutes and 4 seconds. But for now, that covers the signaling step of termination. So before we go off and end the video, I've just made this little diagram to recap what we've learned today. So first is production. Your phenylalanine is a chemical in your blood, which is turned into tyrosine, transported into your neuron, turned into L-DOPA, which is decarboxylated and turned into dopamine. And then dopamine is packaged inside your vesicle. Then dopamine can be turned into norepinephrine, depending if whether your neuron is a dopaminergic neuron, then the pathway will stop here, or if it's a noradrenergic neuron, then it will continue, and dopamine will be turned into norepinephrine. But after you have norepinephrine, then you must release it. So your VAMP proteins and your SNAP proteins, they can interact and link up together, and then your vesicle membrane and cell membrane can fuse together, and your norepinephrine can be released outside your neuron into your synapse. Now, depending on whether your norepinephrine just needs to go across a short distance or a long distance, if it needs to go over a long distance, then it will go over to the adrenal medulla, get a methyl group, and turn into epinephrine, which is more stable and it can travel through the bloodstream, travel further distances, whereas norepinephrine is more designed for shorter distances and it just goes across synapses. And then they can both go and bind to your receptors, so alpha-1 receptors, which are linked to GQ proteins, alpha-2 receptors, which are linked to GI proteins, and beta receptors, which are linked to GS proteins. And GQ proteins can control your enzyme phospholipase C to make IP3 and DAG, whereas GI and GS proteins, they modulate activity of adenyl cyclase enzyme to control levels of CAMP production. And then finally, to prevent responses from going out of control, you have to terminate them. So enzymes like MAO or COMT can come along and degrade and terminate your norepinephrine or epinephrine, or your norepinephrine can be reuptaken into neuronal cells or other non-neuronal cells such as other random tissues, or your receptors themselves can be desensitized by getting pulled into the cell and stored for later. So there's different types of termination mechanisms that prevent these responses from going out of control. But that's the general pathway of norepinephrine and epinephrine. So that basically wraps up today's video. I hope that this video was able to help you better understand the production, packaging, release, response, and termination steps for epinephrine and norepinephrine, as well as help you understand the slight similarities and differences between epinephrine and norepinephrine. But yeah, that about wraps it up. If there's anything that you would like me to make a video on or explain again, or any tips or suggestions on how to improve, I'd really appreciate it. I would read any feedback and any comments that you guys post, so I really appreciate that. But other than that, thank you guys so much for watching. Thank you for all the support, and I'll see you guys next time. Next video, I'm thinking I might make a video about the parasympathetic and sympathetic nervous systems. Not confirmed yet, but we'll see. Until then, thank you guys again for watching, and I'll see y'all next time. Easy.